In this video, we will discuss about a graphical abstract that describes about how one can use this Urocanase positive skin bacteria to help to optimize the immune response. So, what they actually did in the paper is shown in this graphical abstract is that first they took the mice model and allow them to go under this UVB, and thereafter they take a skin swab and do the RNA analysis, the 16S RNA analysis at 8 hours and 24 hours. After that they are able to see the some of the altered bacterial species, and this in this altered bacterial species they are able to find out that it's this urocanase that can helps in the immune response. And there is a process that occurs within this skin also, and for example this metabolism of this urocanase to helps the bacteria in proliferations. So if someone can inhibit those metabolism of this cis uh, UCA formations, then this urocanase can be manipulated to form a immune response, manipulated to control the immune response, and that can in return can be utilized. This strategy finally can be utilized to avoid or prevent the skin cancers in the patients. And here is the final outcome, that alterations in the cis UCA levels that can be done using these inhibitors, urocanase inhibitors. For example, this they have used the gly glycyl glycine. It can prevent the metabolism of this urocanase to cis UCA, and thereby they can fine-tune the immune response and tolerance to environmental antigens. So in this case the cis UCA plays a major role, that its metabolisms needs to be controlled in a way that a person requires immune response on their skin. So if the person is exposed to more of these environmental antigens that can damage their skins, then they can use or accordingly use this glycyl glycine in a way to maintain this urocanase, and that helps in the immune immune response, that helps in the immune response on the skins against the harmful environmental antigens. And these things has been identified, or these experiments are being done on the mice, and so this strategy can, or the results of this paper can be used as a first step towards developing this towards developing a strategy to optimize immune response on the skins of the patients. And there could be many ways to control this urocanase metabolism, or the metabolism of this cis UCA. One of them ways is shown here is glycyl glycine, and also they did this 16S RNA analysis at different time intervals to show that these levels of this urocanase either degrading or they are maintaining at the same levels or are increasing. So this RNA analysis help us to find out the different time interval, and RNA analysis helps the researchers to find out the change in the urocanase and the immune response and its effect on the immune response. We will discuss all the experiments that are presented in this presented in this paper in detail. So for today is just a graphical abstract to get an overview what we are going to discuss in our upcoming videos. That's all for this video. If you need to understand more about their graphical abstracts or read or wants to read the paper in detail, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.